Hello, and thank you for joining Dr. Raux and I for this webinar. Dr. Raux and I are both infectious disease and antimicrobial stewardship pharmacists, and we serve as medical science liaisons at bioMaryU. During this education session, we will discuss clinical perspectives of updating breakpoints. I personally believe that updating breakpoints is an essential step in the fight against antimicrobial resistance, or AMR. The current breakpoints landscape is multifaceted. Some labs are using obsolete breakpoints. There are new regulatory requirements from CAP, and the players in this space, the FDA, CLSI, and manufacturers, are not always aligned. The challenge is many labs and stewardship programs may not be equipped to tackle the task of updating breakpoints. So the question becomes, what support and resources do labs need to update their breakpoints? As a company, we have attempted to understand this. Today, we would like you to understand the clinical implications of updating breakpoints and become aware of the resources that we will be offering our customers and they're out there for you to use as a lab. Using these resources will provide three key benefits. One, it will offer guidance specific to VITEC 2. Two, you'll be able to leverage some of the templates to seamlessly update your lab procedures. And three, it'll keep all of us in the fight against AMR. We do have to disclose that we are both medical science liaisons for bioMaryU. And this is the fourth webinar in this series, 4 of 4. And again, these webinars will be available for you to view on demand if you weren't able to participate in them live. They can be viewed in any order, but we do recommend viewing them in order, particularly maybe this one last, just for the perspective that they really do build up to the point of using the breakpoints clinically. Today, our objectives are to understand the action that we are called to do, discuss the methods to complete the task at hand, and review the results we can expect. And just a reminder, the reason that we're doing all of this, it's not to give more tasks to our labs and to stress all of us out. It really is for AMR. So first, let's discuss our call to action. We'll cover breakpoints in the context of antimicrobial resistance, CLSI's cyclic process for updating breakpoints, a publication that prompted the change among many other things, but this one in particular sticks out to me, and the new CAP requirements that are going into effect in January. So, antimicrobial resistance is a global issue, and it's global because these organisms, these resistance mechanisms know no boundaries. In order for us to be part of the conversation, part of the solution, we must first be able to detect and interpret resistance in order to respond to it. Herein lies the great challenge. Breakpoints are a moving target. The interpretation of MICs via breakpoints continues to evolve, and that's in response to emerging resistance from these pathogens, improving our understanding of pharmacokinetics and pharmacodynamics and the drugs that we're using to treat these organisms, and then new clinical data that are showing differences in outcomes, and maybe that our breakpoints aren't set at the current best threshold. The revolving door of updating breakpoints presents a challenge to labs, particularly those who have limited resources and expertise. And some of those resources could be time or personnel, and always being called back to the bench to be taking care of the patient samples that are at hand. So we completely understand that labs are challenged in this space. However, we also need to recognize that labs that do not apply up-to-date breakpoints jeopardize safe and effective treatment for the patients in front of them, and they also impede global efforts to address this AMR crisis. So as a global leader in infectious diseases diagnostics, bioMaryU has a duty to support our customers through this journey. The process of updating breakpoints, it is quite a journey to update a breakpoint, and it all starts with CLSI. I am personally very grateful that they have a committee that is constantly monitoring this data. So we mentioned it previously, the clinical outcomes, pharmacokinetics and pharmacodynamics, new mechanisms of resistance that are emerging. And their subcommittee is constantly evaluating this data that's coming out. If their committee so decides, they can adjust the breakpoints or the interpretations of those MICs. This information is published annually in the M100. The M100 is an amazing resource. It is available for free, and then you can also get print and PDF versions at a small charge. The CLSI annually reviews, publishes, and then discusses these breakpoints. So in the beginning of the M100 document, there's a review of changes since the last time it was published, as well as a table that summarizes all of those changes since 2010. They also have webinars to discuss these changes, and then it also presents an opportunity to ask questions. So to fully understand how many labs are updating breakpoints, they sent out a group of clinicians and microbiologists sent out a survey to labs in 2019. This questionnaire was incorporated into a CAP survey, and it went to about 2,300 labs. About 1,500 of them responded. They were asked if they were using current or obsolete breakpoints for 7 bug-drug combinations. In the 1,500 that responded, about 1,250 were in the US, and the rest were outside of the US at international sites. The biggest takeaway that I think, one of the biggest takeaways from this paper is that between 40 and 70% of labs, depending on the bug-drug combination, are reporting obsolete breakpoints. 40 to 70%. The kicker is that about 55% of those labs indicated they did not have plans in place to update them. A little bit more information on that, specifically to take a look at the reasons for not updating breakpoints. Manufacturer-related issues topped the list, as did resource limitations. So as a result of this, CAP decided to make a change. And this is where the new breakpoint requirements come into play. So many labs are accredited and surveyed by the College of American Pathologists, or CAP. And the criteria for accreditation is regularly updated. But certainly the most relevant change, and one that has put some stress on our labs, is in the context of breakpoints. So first, labs must establish their current breakpoints in use. And we do have tools to help labs do this, and there are previous webinars on it, and we'll be able to share more of that information as it becomes available. And then labs also, once those the breakpoints are established, they have to understand, or they have to come up with a plan. Excuse me. Once the breakpoints in use are established, they have to come up with a plan to update those breakpoints. So once the breakpoints are updated on the instrument, we have to ask, what additional steps need to be taken? Will the LIS or EMR need to be updated? What are the clinical implications of updating these breakpoints? So to answer this, I will turn it over to Dr. Raux.
Thank you so much, Lauren, for laying the groundwork on our call to action and why this is so important. We will now move into the methods section of our presentation, where we're going to first discuss the importance of the lab-clinician collaboration. Second, we'll deliver some guidance on using susceptible dose-dependent, or SDD. And lastly, we'll disseminate resources, particularly for use in some select populations. It's critical that labs have a collaborative relationship with the clinicians at their institution. The lab director or another representative should be a key member in that institution or health system's antimicrobial stewardship program. When thinking about it with that lens, it might be easier to see some of the ways that this relationship can manifest itself. The lab has the strongest understanding of the local epidemiology. They know what their local resistance patterns look like. The clinician half of the team could then take that information and, using the formulary of the hospital, ensure that appropriate antibiotics are included in treatment guidelines, and ultimately will treat patients with the correct antibiotics. As resistance changes over time, the lab, in conjunction with the clinical team, is able to guide the hospital's pharmacy and therapeutics committee to change what antibiotics are regularly available for use at the institution. And doing testing is only part of the work. If the clinicians can't access or understand the results that are being put out, it may make all of the work that the lab is doing seem trivial. Providing and accepting feedback on how things are reported is critical, and it's a two-way highway that needs constant attention, particularly with some of the changes that we have been and will discuss, things like SDD. Modifying the delivery of that information can absolutely influence the way patients receive their care. People need to hear something several times before it sticks. Having a unified voice across both the lab and clinical teams, antimicrobial stewardship teams, when delivering education, and whether that's education to the house staff or other treatment members, administration, whoever it may be, that's going to lead to more effective rollouts. When the time comes to select a new AST card, if that's something that your hospital is going to be undergoing, let's circle right back to the first point. Making sure that the options that are on the card align with the local epidemiology of your institution. This will also set up that the drugs that the clinical team are used to using are still going to be on that card if that's appropriate. And if it's not a perfect match, which rarely will it be, some concessions are going to have to be made. But understanding, again, that local epidemiology and the institution's formulary, how the interplay is very important. So one very specific and one very significant change that's been put out by the CLSI in relatively recent years is the interpretive category of susceptible dose dependent. So historically, breakpoints have been applied based on the numerical MIC of an organism, and that organism was then assigned a categorical, susceptible, intermediate, or resistant. Clinicians had a pretty good idea of what susceptible and resistant meant. It was probably going to work or probably wasn't going to work, respectively. But there's a lot of confusion about the intermediate category. So why was susceptible dose dependent created? Because of that annual review that Lauren was talking about with the CLSI, where they're looking at clinical outcomes, pharmacokinetics, and pharmacodynamics, looking at new resistance mechanisms that are emerging for different bug-drug combinations, as the microbiology community at large learns more about microbes and antibiotics, there's going to need to enhance susceptibility reporting to be more specific for patients. So ultimately, that led to this fourth category being created, which is susceptible dose dependent. I would like to specifically note that this is only a category that's recognized by the CLSI. At this point in time, the FDA does not recognize this category. What susceptible dose dependent does for the lab, for clinicians, for patients, is it allows the report to highlight a way to showcase that a higher drug exposure at the site of infection for that organism drug combination exists. So that can be achieved by giving a higher dose of a drug than what may normally be given, or providing some alternative dosing recommendations. So that could be a shorter dosing interval, giving it more frequently, extending the infusion of a given antibiotic, or potentially even giving it continuously 24 hours a day. STD was first successfully applied to antifungals and has been transitioned over to bacteria in recent years. Antimicrobial stewardship programs are going to be a great resource for implementing and maintaining STD efforts. Just as clinicians didn't necessarily have a very strong grasp on what intermediate meant or was, they need to be taught what STD is as well. A lab is going to need to determine if they're going to report STD, and if they do, how are they going to do so? There can be some issues with your LIS connecting to the electronic medical record and making sure that it's actually feasible. how you're going to actually be able to do that. Every lab is going to have to look at their individual patient population, the expertise that's there both in and outside of the lab, and discuss with the clinical team. So there are going to be 3 different ways that a lab could choose to implement SDD. So if they're going to align with the CLSI breakpoints that include an SDD, they could report the organism as it was tested and release SDD. They could suppress the SDD result and not release it at all, or they could change it to be listed as intermediate. I believe it's Appendix F in the most recent M100 has some additional recommendations related to that. It also includes some recommendations about comments that could be added to your susceptibility reports to make it a little more clear what the SDD value really means. What it does not mean is it does not replace intermediate. There are only select bug-drug combinations where SDD is currently applicable. If a lab does continue to report something as intermediate and not SDD, what they may want to do, excuse me, is include some additional comments about what it could mean in the context of that culture result, provide education to clinicians on how to either navigate the CLSI M100 so they could understand what that is. And if you need some additional guidance, we at US Medical Affairs are also happy to help with these discussions. I'd like to quickly showcase, we're not going to go through this whole table, but these are the current bug-drug combinations that have an SDD classification per the CLSI. And so quickly, let's just look at an example. So let's look at cefepime in combination with the Enterobacterales. The susceptible breakpoint, which is set at an MIC of 2 or less than or equal to 2, is based on a dose of 1 gram of cefepime administered every 12 hours. SDD has a range of 4 to 8. And the breakpoint at 4 is based on 1 gram of cefepime given every 8 hours, or 2 grams of cefepime given every 12. Lastly, the MIC of 8 is based on the max dose of cefepime you can give a day, which is 2 grams administered every 8 hours. So as you can see, the dose changed based on the numerical MIC value. A clinician could optimize the patient's treatment regimen to have the least amount of drug administered on a daily basis, hopefully going to decrease some adverse outcomes and potentially save some costs. We wanted to highlight a few resources for pediatric populations. We're not going to delve into any of the data here today, but there are several publications that have come out over the last several years if your institution serves a pediatric population to really consider what the role of SDD in that patient population is as well. So lastly, by supporting our labs on this whole journey that we've gone on today, and providing tools that we hope will be of benefit for use. We are providing benefits to patients, hospitals, and the world. To the patient, we're hoping to give the correct antibiotic at the correct dose and at the right time, and hoping to improve their clinical outcomes. For the hospital or lab, we're hoping to assist with CAP compliance, allow you to be able to provide reliable and updated results, building trust amongst the community, and improve empirical therapy for your patients. For the world, for the globe, there are potential benefits from a One Health perspective and making progress here. The updates here may increase your external validity for surveillance reporting. And finally, to bioMérieux for us. So this fulfills our mission to improve public health worldwide. We want to be providing consistent messaging and have satisfied customers and build trust, again, along that pathway. For me as an employee, for I think many of the folks that are working on this, this is very satisfying. It's fulfilling our duty as a major provider of automated susceptibility testing to cascade up what else is shown here on the slide. So in closing here, I will move us back over to Lauren.
Thank you, Brian. And I couldn't agree more. I think that we are both very honored to be part of the task force that bioMaryU has put together. This is an immense passion of ours. For me personally, stewardship is why I do what I do every day, so that we can have antibiotics for generations to come. And the more effectively we can use our antibiotics for these the organisms and their mounting resistance mechanisms, I think the better off that we're going to be. So lastly, we're just going to go through some resources. So the concept of susceptible dose dependent is pretty broad, and there's a lot of intricacies within it. Excuse me. So we do want to provide some resources here, and at the end, we'll talk about some ways that you can contact us at bioMaryU to talk about some of the specifics of your lab. So we've mentioned the FDA STIC website in some of the other webinars. This is a great resource. It's aligned based on drug. I personally have it saved in my favorites because I visit it so often. And they, again, to recognize that the FDA does not recognize the susceptible dose dependent category, you'll see everything there as intermediate. The CLSI M100 document with, we've mentioned it a few times now, as well as the education webinars that CLSI puts out. Within the M100 specifically, there are 2 appendices related to susceptible dose dependent. Appendix E provides some of that drug dosing. Be aware that a lot of the dosing in there is for patients that have normal renal function. And if there are any modifications that need to be, there are other resources for that, but it's certainly a starting point. And then Appendix F is kind of a Q&A, frequently asked questions type section. And those webinars, there are definitely some previous webinars too that have some information as CLSI continues to provide that information for, or that category for more bug-drug combinations. Some of these publications, in addition to the pediatric ones that Brian presented, the primer for clinical laboratories on updating breakpoints by Humphreys and colleagues, I mentioned it in the previous presentation. One of my favorites to go back to and provide some really great guidance on how to approach this. And I believe that there actually is information on SDD in that publication as well. Similarly, that second publication by Humphreys. And then lastly, a publication from Dr. Prinzi on antimicrobial susceptibility testing. All 3 of these I keep close in my favorites or in my frequently used documents, just because I do reference them so often and share them with colleagues and customers. We had a webinar, webinars number 2 and 3 really go over a lot of the bioMaryU specific resources that we've put together and want to provide to our customers. All of it will be housed on a microsite. So education is something that is really important to us, both for our internal employees and for our customers. So that's why we decided to put together so many live presentations for you today. We also have the Breakpoint audit tool. So we have the BAT, and then CLSI is formulating what they call the BIT. So there will be other resources coming from CLSI on establishing current breakpoints in use and updating them. Our BAT tool is going to be specific to Vitech. However, it will be an opportunity and a way for you to actually update it with other methodologies such as disk diffusion and e-test if you use those methods in your lab. So it can be a comprehensive document. And we'll have instructions for use on that and more webinars when those documents become available in the coming weeks. We also have that FAQ document. There is a whole section on susceptible dose dependent that you can refer to and use it to actually work with your clinicians if you're in the laboratory or vice versa. And then the verification and validation documents, we've gotten some feedback on these already that they are incredibly helpful for walking through the process of performing a verification. Again, reminder, that is when the breakpoint was set by CLSI, recognized by the FDA, and then received FDA approval for here at bioMaryU or whichever manufacturer you use. So that's verification. Validation is when there's a discrepancy somewhere in there. Maybe FDA doesn't recognize or we haven't received the approval at bioMaryU. So both of those documents will be there for you to use. And that concludes the clinical perspective of updating breakpoints. Again, you can find more information on what bioMérieux is doing specifically in webinars 2 and 3, and then general information with Dr. Princey on updating breakpoints in webinar number 1. These are all recorded and will be available on the Labroots website. Please don't hesitate to reach out to bioMérieux. We're putting this information out there. We're providing resources and education and tools because we care. We truly believe in that we have to stay in this fight against AMR. I think some of us also understand that it's often that silent pandemic that we're not necessarily paying attention to right now. But the more we can do proactively, I think the better off we're going to be. If you have any questions, there is no Q&A with this, but if you do have any questions, please put them in the chat. They will come to us and we will respond directly to you via email. Other resources at bioMérieux do include your sales representative, your field application specialist. We have our call center, and we also have our medical affairs, medical science liaison team. So please don't be a stranger. We're here to help you in any way that you can. Thank you so much for your time.